This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
The Science Notes podcast on OAR FM Dunedin, brought to you by the Division of Sciences at the University of Otago. I'm Dave McMorrin. Over the next half hour, I'll be chatting with a graduate science student about their research, their passion for science, and the things that fuel their creativity. Well, hello there and welcome to another episode of Science Notes. My name's Dave McMorrin, and this time our guest is Lily Bentall. Hi, Lily.
Good morning.
Thanks for coming along.
No, thank you for having me.
On a coldish Dunedin morning in the winter.
Yeah. It's tough getting out of bed this morning.
I'm sorry. Lily is doing a PhD in anatomy.
Neuroscience, actually.
In neuroscience,
in the
strictly.
Department of Anatomy.
There you are, neuroscience in anatomy. So, neuroscience, brain science.
Yeah, brain science.
Regular listeners might realise that the previous guest we had on the show was also doing, he was doing brain science, sort of. He was interested in intracranial pressure.
Oh, interesting.
So he's interested in what's going on the outside of the brain. You're interested in what's going on the inside.
Yeah, really deep in the brain.
Way in the middle.
Yeah.
So before we get into the nuts and bolts of that, how is it that you find yourself doing a PhD in neuroscience?
Yeah, it's funny. I came to Otago. I came to Otago in 2017, and I just did first year health science.
As you do.
As you do.
I apologise.
Yeah, I actually think you lectured me, to be honest. So I came from a really small town, and I thought, okay, I'm good at science.
What is your small town? Shout out for
Levin.
Levin,
yeah. An hour above
I know
Wellington.
Levin.
Yeah.
Yes.
So I thought, okay, I'm good at science. That must mean I have to do medicine, or that must, that means that's the direction to go in. And I came and I did first year health science. I did Hubs 191, as everyone does, and there was maybe 5 lectures on the brain. And I just thought, wow, that just clicked for me.
Yep.
And it's interesting, neuroscience, it's you either love it or you hate it. And I had so many friends that hated it. They just wanted to get through those classes, the test questions as fast as they could. But for me, I wanted to know more. So after the first year of uni, I switched my major to neuroscience. And the rest is history. I just loved it. I thought, even we have a lot of fully anatomy papers where you learn about every structure in the brain, and it all just lit me up, and I thought it was really interesting. I just remembered everything really easily, and I thought that must be a sign that I'm on the right track. Mm-hmm. So I just kept going. And then in third year, you start to think about what you're going to do next. And I met my supervisor. She was lecturing, talking about Parkinson's disease, and the pathway in the brain that's involved in movement is just really interesting to me. And so I interviewed with her, got my honours project, and then continued on with the PhD. Right.
So you've been kind of doing the same thing all the way through?
Yeah, but I think it was interesting. I joined the honours at a really great time. She was just finishing a massive HRC grant, and she was using optogenetic stimulation, if you've heard of it, where you implant a viral vector into the brain, and it makes that neuron responsive to light. Right. And so you can put these promoters in that make it only express in really specific neurons. Like electrical stimulation, you stimulate everything in the surrounding area. With this, you can. We made it really specific to a certain type of neuron in a certain part of the brain, and then we stimulated that part of the brain.
Right, just by shining light.
By shining light.
Rather than electrodes and wires and.
Yeah, so we just put an optic cable into the brain.
Sure.
And so these animals were Parkinsonian, so we induce Parkinson's disease in the brain using a cranial surgery called stereotactic surgery. And then these animals, they do a reaching test, very similar to humans. They reach, and then we applied optogenetic stimulation, and that recovered their reaching. So when they were Parkinsonian, they lost it, and then this optogenetic stimulation recovered it. And so that was my honours, which was a pretty crazy honours project, really.
If I'm keeping track, was that 2020?
2020,
Wasn't a good year for doing your first year of research?
It was really intense. I think I'm really lucky in anatomy. They structure it. All of your papers are in the first maybe 4 months.
Right.
And
Which
so,
is when all the lockdowns were.
Yeah, so I just spent all of lockdown writing my literature review and doing my papers. And I was actually an RA at the time. I was at Cumberland College. So I stayed at Cumberland College and I was just sitting...
And it'll be nice and quiet because there wouldn't have been as many students
Yeah, I think there was like 50 of us altogether out of 350. So it was really fun, got a nice group of kids and I could just really focus. And then as soon as we were allowed back in the lab, it was like 7 days a week, just giving these animals Cocoa Pops so that they could reach. And that was I just thought it was so interesting. And my dad's a vet, and so the animal work was quite normal to me. I felt like...
Yeah,
I could really read them quite well. And
is this a thing? I mean, this is kind of slightly off topic, but given how much animal work that you do, and given that I'm guessing it's not for everyone, does this kind of a sort of a separation point in neuroscience students as they go through, the ones who are happy with the animal stuff and go one way, and the ones who aren't and go a different way? Definitely,
and there's such different. Like neuroscience is so diverse. You can do cell culture where you're in a different type of lab and you're feeding cells, and then you can do, I don't know, bioinformatics or something like that, and then you can do animal work. And there's a lot of students that start animal work straight away. No, I don't want this. So I would really recommend doing that in your honours to see if that's something you could do, because if you can't do it in your honours, there's going to be a PhD student or there's going to be a postdoc that can help you and you can get through. But I think really starting a PhD with animal work, you have to be okay with it.
So you're studying Parkinson's?
Yes.
And your notes that you've sent me said that it's the second most common neurodegenerative disorder in the world. I think probably everybody knows someone who knows someone at least who's had Parkinson's. My grandmother had Parkinson's. Do we know why it's so widespread? Why it's such an issue?
I think that's the $1 million question. But there's definitely risk factors. So it's idiopathic. So there's maybe majority of it has no genetic cause. There's early onset Parkinson's that has a high genetic correlation, but majority of people get it past the age of 60. That's what we call late onset or regular Parkinson's, I guess you could say. So there's a few risk factors. Sort of pesticides can be really bad. So they found people living around agriculture or around vineyards quite often would get it. But really it's just idiopathic. And I think it's really to do with the fact that dopamine neurons, when you lose your dopamine neurons, you get Parkinson's disease. These neurons are really susceptible to dying, essentially. There's something different in them. They're
not as resilient as some.
Yeah, exactly. And so you lose them, and then you start to develop these really horrible effects.
And unlike a lot of diseases, it seems to me, there is a drug which everybody has heard of. Everyone's heard of L-Dopa, I think. Yeah. And it says still the drug that is used.
I think it's really interesting, and I think that's... So I talked about my honours project, but then when I came to my PhD, Louise said to me that I don't really have a project for you. Think about what you want to do. And so I took a year off, and I was researching Parkinson's, and I looked at levodopa, and I was like, wow, this is the drug, but why does 80% of patients develop this side effect, and why are they still giving this drug if that happens?
Yes.
So this drug was developed in the 60s, and it was when they found it, wow, incredible, this has revolutionised the landscape. And then about 10 years later, all of these patients started developing this side effect, and that's when they sort of realised that this was going to happen, and they had no other alternative.
And still,
yeah. The alternative is now deep brain stimulation, but I think we only do 10 a year in New Zealand.
So that's actually electrodes into your brain to stimulate things.
And there's so many exclusion criteria for that. You can't have it when you're too old. It costs a lot of money. You can't have depression and all those other sort of things. And it can affect your speech because placing of the electrode can be really dangerous. So essentially, when you get Parkinson's disease, what they do now is you get given physio, and so you're trying to keep moving just so that you can prolong the period where you don't need to be dosed, and then you get given levodopa. And you've got about 5 to 10 years of really good outcomes, and then you start to develop this side effect called levodopa-induced dyskinesia.
Yeah.
And so once someone develops that, their life sort of becomes this flip-flop where they wake up and they have, the drug isn't in their system, and so they struggle to move, like the classical sort of Parkinsonian symptoms, and then you take the drug and you might have a good grace period where you're normal like me and you, and then they develop this side effect where it's actually the opposite end of the spectrum and they can't stop moving.
Right.
And I find that quite often in sort of layman's terms, people get those symptoms mixed up. If you saw someone with Parkinson's disease and they were sort of flailing about on their chair, some people think that is Parkinson's, but that's actually dyskinesia.
Right.
So Parkinson's is more sort of being quite still and maybe a small tremor in the hand.
Right.
But these large movements, that's dyskinesia.
Right. And that's the treatment causing that.
Exactly.
Right.
And I've chatted to a few people that have had this dyskinesia, and they said that yes, they have this grace period, but there's that real anxiety around it, that I have to get everything done in these 2 hours.
Yep.
I have to have my whole day done so that I can go home and experience these symptoms in the comfort of my own home. Because if I'm walking down the street, someone's going to look at me strange I've heard those stories and I thought, oh my gosh, this is where I want to go.
Yeah.
It's in Parkinson's, it's in the movement part of the brain, but for me that really lit me up, and that's where I started with thinking about my PhD, and so it's something that I feel very passionate about.
As is clear.
Yeah.
So you're specifically then interested in exploring a particular part of the brain, which up till now has kind of been neglected as being of importance, but your lab disagrees and thinks it is important.
Yeah. So my PhD, Parkinson's disease is really important in the basal ganglia. I think a lot of people have heard about that part of the brain. It's these group of nuclei that are important in initiating movement. And anyway, they go downstream to this place called the motor thalamus. And quite historically, the motor thalamus, or the thalamus as a whole, has really just been thought of as a relay station. So when we have any sort of feeling in our fingers or we move our fingers, those pathways go through the thalamus. And so that was sort of just thought of as a relay station. And my supervisor did her PhD on the motor thalamus and found just completely that wasn't true, that the motor thalamus is actually really important in movement, and it receives inputs from the basal ganglia. It receives input from the cerebellum as well, which is also important in movement, and then it receives information from the brainstem and other areas. And what she hypothesizes what we hypothesize is that it takes all this information and it synthesizes it and projects it to the motor cortex. It's sort of like the teacher, getting all these inputs and realizing what it is and pushing it up. It's because of that, it's been really ignored. Majority of the papers just look at the basal ganglia or look at the motor cortex, and they don't look at the thing in the middle. So Louise found lots of really cool papers, lots of really cool research showing that the firing rate changes, that the type of burstiness changes, but no one had looked at it in dyskinesia.
Right.
Yeah, so that's where I sort of came in. And so I went a step further and I had these Parkinsonian animals and then gave them induced dyskinesia and then recorded in the motor thalamus.
So do you give them L-dopa?
Yes, yeah, right. Just like a human.
So just like that, yeah.
And then they developed the symptoms very similarly. And I actually changed the dosing rate so that I was trying to get 50-50 split of animals that had dyskinesia and animals that didn't. And that was sort of to look at, so I had 4 treatment groups. I had my controls, presumably their brain should be acting normally. You had the Parkinsonian animals, which we already knew how their brains had changed. And then we had our dyskinetic and our non-dyskinetic animals. And technically, we were hoping that the non-dyskinetic animals would show a Parkinsonian human when levodopa is being therapeutic. And then the dyskinetic animals were showing when it was pathological. And And so I recorded in the motor thalamus, but I also recorded upstream in one of the basal ganglia nuclei as well, which has been explored to death in this landscape. And then that meant that I could really show my skills. Okay, yes, we found the same changes as previous. Now look what else we've found. And actually, the changes here are really significant and quite compelling. And so that was a big part of my PhD, and now I'm just trying to write up the paper and get it submitted.
So, in your day-to-day then, what you need to do is to put electrodes into these very specific parts of the brains of your little animals that you're looking at.
Mm-hmm.
This was a note I made when I was just reading through this before. Mm-hmm. But people talk about the brain as having all these different little regions.
Yeah.
Right? And it seems like that every time you look at another paper, there's another weird Latin-named region in your brain. How do you distinguish one from the other?
Yeah, so there's actually these things called rat brain atlases or mouse brain atlases.
Right.
And the same for the humans. I think there's now one for a sheep, one for apes and all those sorts of things. So, and they're based off sutures on the brain. And so we would open up the skull and we would say, okay, here's a suture here and there's the other suture. You do some mathematical equation that at this certain coordinate will be my nucleus of interest.
Okay. And the brains are similar enough from creature from individual to individual to individual
similar
that
enough,
this works.
Yeah.
Right.
And one thing we did actually, so we were the nucleus that was in the basal ganglia was really, really deep, and above it is a set of dopamine neurons. And so what we would do is we'd go down and we'd sit and then we'd find those dopamine neurons. We didn't want to record them, but we'd find them and we'd go, okay, now we've found them, so we know that perhaps 100 microns below this is my area of interest, and I would only record from that area. So you can do it. It's actually, I think electrophysiology is so interesting because you're recording these neurons and you can sort of estimate that you've got the right neuron by the shape of their waveform. So dopamine neurons are really big, and so they have their waveforms are really fat and really long. And then you go and find that my neurons of interest, which are GABAergic or inhibitory neurons, and they're really, really tiny.
Right.
And also one really cool thing is that you can electrophysiologists, they transfer that waveform into sound. So we use an audiometer. Mm-hmm. And it's so interesting because you'll be going boof, 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 and that's a dopamine neuron, and then tink, tink, tink is my GABAergic neuron. And so, and my supervisor said to me, like, just wait till you become an electrophysiologist because you'll just know when it is.
Yep.
And so those are the sorts of things. And then that's while you're recording the neurons. And then when you finish recording, you inject with iontophoresis, with a current, you inject some blue dye. So you can look exactly where the last recording site was. So you can then perfuse the animal and section the brain. And then from there you can do some maths to find out where every recording site was, and you chuck out any recordings that were outside of your area of interest. So you can be like 99% sure that every neuron is the neuron you want. Right. Yeah. So it's
a thing.
It's really, really precise, and it's long days. Like when I did those recordings, so I had two electrodes in the brain. So I would go and find one, and then I would switch my channels, and I would find another, record them for 5 minutes, and I'd be in this dark room for like 12 hours. And I remember reading once that you're in the right job even when there's a day like that, and you walk out and you feel happy. Like, some of those days were really hard, but I would leave at 8 p.m. and I'd be like, that was awesome. Like, I got to record single neurons today. Like, I'm so happy. And so, I did that for 50 animals, and that was my PhD. Mm-hmm.
One of the things that you've noted in your notes is this difference between the effect of this side effect, if you like, of the L-DOPA. The difference between men and women.
Yeah.
Do we know why this is?
Yes. So for me, it started so in Parkinson's disease, quite a lot of labs will just use men because there's this real sexual dimorphism. Men are far more likely to develop Parkinson's disease, and so for lab animals, you'll just use men.
Yeah.
But actually, I went into my 6-month meeting and someone on my committee said, "Why are you only using men?" And I said, "Oh, just because that's what everyone else does." And
It's science.
Yeah, and she said, "That's ridiculous. You should go back and think about why." And then I did some more work and I realised that, yes, men are more likely to develop Parkinson's disease, but once you've got this cohort with Parkinson's, actually women are far more likely to develop dyskinesia.
Because they live longer.
Not necessarily, no.
Well, I mean, but that would also be a reason to study because it's going to be more affecting the woman.
Yes, that's true. But actually there's this real interaction between estrogen and dopamine.
Right.
So I as part of my PhD, I went on and I wrote a whole review looking at how estrogen interacts with dopamine. And that was really cool because there's so many papers looking at estrogen in terms of Parkinson's disease, but there was not a single review paper looking at estrogen and dyskinesia.
Right.
So we were showing that, we hypothesized that estrogen, it maintains those dopamine neurons, it protects them, has a neuroprotective effect, but actually we hypothesized that it has a role in maintaining dopamine into the right sort of area. So we say when you give levodopa, you have these large swings of dopamine which cause these large flailing movements. And we think that estrogen, when estrogen is high in the system, it maintains that the dopamine into that sort of Goldilocks zone.
Right.
And when a woman becomes postmenopausal, that's gone. And so those previous neuroprotective systems that were there are gone. This dopamine starts surging really non-physiologically, and then they develop levodopa-induced dyskinesia far more likely than men. So that paper's been published. That was really great. And also we included women in our study. We included postmenopausal animals. And we're now in the stage of looking at all of my results and splitting it further into male and female and finding that similar trends occur, but they're far more intense in women, which is, so essentially, more women are experiencing these side effects far more intensely and far more quickly than men. Mm-hmm.
Another example of where science historically has been a bit rubbish in this.
Yeah, we're not just little men, actually.
No, but actually, if you actually studied the woman as well, then you find actually it's even more important that you understand and you try.
Exactly.
Because it's worse,
Yeah. They show that. So women experience dyskinesia. They experience it. Their symptoms are worse, but they also experience it much quicker. Their time to onset is about 2 years shorter than men. So this review paper that we put out was really looking at the sort of cellular reasons as to why this might be happening, but also quite sort of trying to pick up the conversation of, okay, so these 3 papers point at the prevalence changing, but these papers were published 20 years ago. Come on, clinicians, let's get this going. Because it has to be, in order to work for women, it's going to have to be so many different levels of science working together to understand.
And surely there must be motivation/money involved in the studying the second most important neurodegenerative disease in the world.
Yeah.
Yes.
And especially the side effect of the gold standard. Yeah. Almost every.
Yeah.
Mm-hmm.
We're just about out of time. You're how far through?
I've actually just defended.
Actually, no, you did tell me that too. You actually have just defended, so you're almost Dr. Lily.
Yes, I graduate.
Unofficially Dr. Lily.
Yeah, I graduate in August. So,
cool.
Yeah, really excited. And I'm currently working for my supervisor. I've verbally accepted a job in Germany.
Oh, cool.
Yes. So, hoping.
So more of the same?
No, actually, they look mainly at the visual system, and they use a technique called 2-photon imaging. So, they use microscopes. They make these neurons kind of responsive to light, but essentially they light up when they activate, and so we can look at the whole brain working. Yeah, so it's
right.
Yeah,
so it's a little bit more like your honours, the stuff you were talking about earlier, the optoelectronic stuff.
Yeah, a little bit. Yeah. And so I'm really interested in imaging the brain and whether that's optogenetics, stimulating, and then recording, and then 2-photon imaging. I just want to understand fundamentally how neural circuits work. How do we move? How do we see? So that's where I'm going to head with the next.
Right.
Yeah.
So it's not necessarily Parkinson's.
No. So this one, they're just looking at visual cortex in normal individuals. But I'm really interested. I haven't been given a project by them, so I'm interested to see if we could somehow work together, looking at taking their imaging and moving it up to the motor cortex and looking at how, trying to apply it to the movement pathway, which I think is just interesting.
Excellent. Well, all the best for that.
Thank you so much.
And thank you for coming and talking to us
about
what you've been up to. It's fascinating stuff.
Thank you so much.
Cool. And remains for me to thank you all out there for listening. And we'll see you in the next episode. You've been listening to the Science Notes podcast, brought to you by the Division of Sciences at the University of Otago. To listen again or to find other episodes in this series, go to Otago Access Radio's website, oar.org.nz, or to your favourite podcast platform.
This podcast was produced by OAR FM Dunedin with support from New Zealand On the Air.